Welcome everyone. At this point, we have discussed mitosis and we have discussed meiosis. Both are processes that have some similarities, but are used in entirely for different purposes. So this is going to kind of recap on both of these processes. And as you kind of watch this movie, you're going to have the video stop, questions are going to pop up, and you'll be asked to address those. So feel free to go back and re-watch sections of this if necessary so that you can be successful in your attempts at answering those questions. As you do answer the questions, you will see my responses to those questions, so you can compare what you wrote or typed relative to what I have. Feel free to make any notes on your end that you might want, as you can go back and reference these for future assignments and whatnot. So we're going to start by looking at the process of mitosis, which is illustrated on our screen right now. First and foremost, let's talk about the purpose of mitosis. We start with one cell and we go through the stages and we end up with two daughter cells. These two daughter cells are exact copies of one another, and they are exactly the same as the cell that we started with. So if you compare this one to this one, you'll notice that they're exactly the same, and then if you compare both of these to the original one, we'll notice that they're exactly the same. The process of mitosis happens in a form of cell that we have learned known as somatic cells. Somatic cells are cells of our body. So these would be cells in our skin, in our heart, in our kidneys, in our liver, in our stomach, in our intestines, so on and so forth. If it's a cell in our body, then it's a somatic cell, and those cells go through mitosis for the purpose of creating two new daughter cells that are identical to one another. Typically, this is done as the original cell kind of has gone through its life and is kind of nearing the end, and if it dies, it needs to be replaced, so it kind of goes through this process to replace it. So with that in mind, why do we actually need our cells to go through this process? Well, let's just stick with skin cells since we've been kind of mentioning that already. Your skin cells, they have about a 30-day lifespan, and imagine if this cell, a skin cell, dies and it does not replace itself by going through mitosis. In essence, what we would end up doing is we would be losing skin cells, not replacing them. Eventually, we'd have no skin. So if we took away the process of mitosis, we could not survive. Our cells would live, and then they would die. They wouldn't be replaced. No more cells equals no more us. We also learned about Henrietta Lacks a little bit with the HeLa cells. The cells that she had that were mutated were her somatic cells. So when she would go through the process of mitosis, those cells, they would create more and more of those gelatinous purplish cancer cells. And also we saw all the mutations that kind of went along with them. So since Henrietta Lacks's somatic cells were mutated, she could create more mutated cells herself by this process, but she could not pass that mutation along to her children. Now, why is that? It's because she can't give a skin cell, a heart cell, to a child. Those are not used in the process of reproduction, and since they're not, her children cannot inherit her mutated chromosomes, as we kind of saw from her karyotype. Now taking a look at meiosis. Goes through a similar process as mitosis, but a little bit different. The process of meiosis only happens in germ cells. The term germ cell refers to any cell in our body that can give rise to gametes. So recall gametes are either sperm or egg, depending on male or female. So germ cells would be the cells that create sperm or create eggs. At the beginning of meiosis, we start with one cell, a germ cell that has a total of 46 chromosomes, 23 from each parent. So it is diploid because it has one full set of chromosomes, one from each parent. In essence, the purpose of meiosis is to take this one diploid cell with 46 chromosomes and create 4 cells that are haploid that would have 23 chromosomes each. These gametes that are then produced could be used in the process of reproduction. It's also important to note, as we compare all these gametes, we can see that each of these is a different cell. This one looks like this one like this one looks like this one looks like this. So that means that the genes contained within this cell are unique to this cell and this cell alone. No 2 cells will have the exact same genes as the other. Now, when I say genes, be careful. They will have the same genes, but recall that genes can be expressed in different forms. Hair color can be brown, black, blonde, red, whatever. So they'll all have genes for hair color and everything else, but the variation of that gene is going to be different. This one might be for red hair, this one might code for black hair, blonde hair, and dark brown hair. So different for all. Now, since these cells only would have half of the original number of chromosomes, 46, they're no longer diploid cells, they are haploid cells. And when you combine one haploid cell, like a sperm, with another haploid cell, like an egg, you would have 23 from the sperm, 23 chromosomes from the egg, 23 plus 23 during fertilization would create a new cell, a zygote, with 23 from each parent. It would now be a diploid zygote, and that zygote would go through mitosis at that point to give rise to all the cells that we have in our bodies. Some things to consider during meiosis. We can see that when the homologous chromosomes kind of pair up, which would be in metaphase more specifically, we can see that they do cross over, and that's when sections of the blue chromosome trade with sections of the red chromosome as they are separated during anaphase 1. So we can see part of the blue chromosome attached here, part of the red here. These 2 sections would contain the same genes, but different variations of it, brown hair, black hair. Ultimately, what this does is this is the reason why nobody else will look exactly like you. Where will these 2 chromosomes cross over? Might be up high, might be down low, maybe not even at all. It's 100% random, which is kind of what makes each individual unique, and why I previously mentioned no one will ever look like you. It's a direct result of this crossing over. Now this image, it does simplify this a little bit as it only shows 1 pair of homologous chromosomes, but keep in mind that in our cells we actually have 46 of these that are randomly crossing over here, there, maybe once, maybe twice, maybe 16 times between 1 pair of homologous chromosomes. And again, as previously mentioned, we go from a diploid cell to haploid cell. So essentially what we are doing are reducing the number of chromosomes contained within each cell, which is called reduction. We can see here that we have 2 duplicated chromosomes, but now each of these cells only has 1 duplicated chromosome, and here each of these cells only has 1 individual chromosome, which were chromatids up in the stage previous. So these cells, they are found in one specific place in our body. For males, these germ cells are located in the testes, and that's the only place. They are not considered somatic cells. They are not body cells because they are strictly used for the process of sexual reproduction. For females, the germ cells are located in their ovaries, and they again are not somatic cells because they are not body cells. They are used strictly for the purpose of sexual reproduction. Now, going back to Henrietta Lacks, her children could not inherit her mutations in her chromosomes because her germ cells, they were not mutated. It was strictly her somatic cells. Should a germ cell be mutated, then it could be passed to offspring. That was not the case with Henrietta Lacks. Her germ cells were fine, it was just her somatic cells. So all of her children, they were born disease-free, no mutations, because her germ line was not mutated. This concludes our discussion over mitosis and meiosis. Once again, as the questions come up at the end, you may want to go back and revisit some of the things that we have discussed previously.